interactions takes place and the reaction is carried out. Okay, so enzyme technology is nothing but it is with the application of enzymes as tools of industry, agriculture, and medicine. So now if we see, okay, now we have discussed what is enzymes. So what is enzyme biotechnology? So in the first class itself, we have discussed like what is biotechnology? It is nothing but to alter a process or an organism. So here enzyme is a biological component and we are utilizing that enzyme or the process for applying them in the fields of industry, agriculture, and medicine. So if we want some enzymes which are deficient in the patient, so we can use those enzymes. We can create them artificially and we can induce them for the treatment. So like that. And some enzymes, even in plants, they get involved in some process. So we can use those enzymes to increase the production or speeden up the reactions like that to increase the shelf life. So you have variety of applications. So the value of using enzymes over inorganic catalysts in the technological field is due to their efficiency, selectivity, and specificity. So you might get doubt why we need to do enzymes. So we have some catalysts, other catalysts, which are synthetic ones or inorganic ones, which we can use. But rather than, they will have some side effects. Rather than that, enzymes are natural ones. And if we use them, they are more efficient, they are very selective and very specific, and no side effects are there. So that's why we can use these enzymes. So the value of using enzymes, the enzymes are able to operate a wide range of temperature, atmospheric pressure, and even pH. So the best example I will give you in recent times, whatever we have discussed, during animal cell culture, I told you about the enzymes which help in isolation of cells, which are like for the separation of cells. So do you remember what are the enzymes? Trypsin, pepsin. So these are some enzymes, collagenase. Okay, so these are enzymes which we use in cell culture. So that we can actually produce by using this biotechnology. And we have enzymes which can operate at variety of temperatures. If you are aware, like in PCR, polymerase chain reaction, we have DNA polymerase. So what is that polymerase? There are many, but generally used one is Taq polymerase, TAQ, Taq polymerase. So what is the Taq polymerase is nothing but it is isolated from Thermus aquaticus. That is a bacteria which they have isolated from hot springs. Hot springs are nothing but the water bodies which are very hot. Okay, the temperature of the bodies are very high. So these bacteria can survive in that hot springs. Therefore, the enzymes are capable of sustaining to very high temperatures. So why we have selected that polymerase? Because during PCR reaction, to denature the DNA, we have to give the temperature up to 95 degrees centigrade. If you take normal polymerases from human source or any other source, they will get denatured at that temperatures. So in order to sustain the temperatures, the scientists have isolated the polymerase, DNA polymerase from Thermus aquaticus, and they have named it as Taq polymerase, and that is widely used in the PCR reaction. So these are commercially very important enzymes which are already in use. And enzymes are biodegradable. Okay, so if you don't want to use or if you don't want to further continue, you can easily biodegrade. You can degrade the enzymes rather than if you take the other inorganic or any other catalyst, they can get accumulated in the nature. But because enzymes are of natural source, we can easily degrade them. So these are some of the applications and advantages of using enzymes and catalysts, and therefore enzyme industry. Okay, enzymes use has led to the development of an industry, and therefore enzyme biotechnology branch has been developed. So here, these are some of the enzymes, some of the functions of the enzymes, like in the degradation, in the synthesis, and in the modification. There is catabolism, anabolism, and alteration. Okay. So during cell growth and nutrients of the substrates are converted to cell mass, then they are called primary metabolites. So we have two types of metabolites, are nothing but the products which are generated during the metabolism. So we have primary metabolites and secondary metabolites. Usually secondary metabolites are seen in plants. Again, they are commercially very important. And you have another branch of biotechnology, like in the plant biotechnology, where we exploit the secondary metabolites. But as of now, so what are primary metabolites? They are nothing but primary metabolites are very important for the survival of the organism, whereas secondary metabolites, they just add to the survival, but they are not actually required for the survival. Okay, that means if you remove the secondary metabolites also, the organism will survive. So that is the thing. Okay, so the cell mass itself consists mainly of proteins, but a primary waste products are also formed, like carbon dioxide, lactic acid, ethanol. All these also will be formed as a byproducts. Okay, so along with the cell mass during cell division, even primary metabolites and byproducts also will get generated. So here you can see that this is the photosynthesis. So from photosynthesis, what is photosynthesis? Photosynthesis utilizes the water, light, and carbon dioxide, and it will generate sugars and oxygen. And this oxygen again utilized in the respiration, and that will again produce the water, energy, and carbon dioxide. So during this process, usually we will get all sugars, then fats, proteins, hormones, and all the primary metabolites will be generated. Then we have secondary metabolites. So secondary metabolites are not directly related to the cell growth, but they are the side products of bacterial life. They are produced in the low concentration, but now in the laboratory, because of some mutations, and scientists are trying to overproduce this metabolites. Okay, secondary metabolites. So here, many antibiotics and vitamins are secondary metabolites. So we don't require antibiotics or vitamins in large quantities, right? We require them only in the small quantities. And those are, again, they are part of the metabolism, but they are not actually required for that particular organism where they are generated. And the formation of secondary metabolites is not directly proportional to the primary metabolism and cell growth. That means, though if primary metabolites are in high concentration, but the secondary metabolites may be in low concentration. It is not proportional. Okay. So these are some of the examples of secondary metabolites, like pigments. In carrot, we have carotenoids, then anthocyanins, then morphine, or then diterpenes. These are in plants, rubbers, gums, cellulose. So many other secondary metabolites are there. Now, what our enzyme biotechnology is actually doing is, so during the production of this primary metabolism or primary metabolites or secondary metabolites, we have some enzymes which are involved. So now what we are doing, by increasing the production of enzymes, we are again increasing the production of the product also. Okay. And another thing is, from enzyme technology, we have already got some products. So what are they? Protease, which is found in papaya. So this enzyme can act as meat tenderizer. It can tenderize the meat. Okay, so therefore, it is again applicable. So whatever the enzymes which we are generating in enzyme biotechnology, that enzyme can be applicable in another industry. So microorganisms have been used for thousands of years for making wine, beer, vinegar. So through fermentation, many products have come. So these microorganisms are used as a source of enzymes during the production of the products of biotechnology. Now here, many industrial processes now make use of pure sources of enzymes. That is, the enzymes have been isolated from the microorganisms before use. So they are nothing but purely, they are not synthetic ones. So what they are doing, they are growing the bacteria, and from that, the expression of enzymes have been increased, and from that, these enzymes are extracted. So they are mainly from the pure source rather than synthetic ones. So what is the structure of enzyme? So before going into the technological part, first we should know what is the structure of the enzyme and what are the components. So enzymes have non-protein part and a protein part. Okay, and the protein part is called as apoenzyme, and we have non-protein part. So non-protein part again can be a coenzyme or a cofactor. If a mineral is involved as a non-protein part, then that is called as a cofactor. If a vitamin is involved as a non-protein part, then it's called coenzyme. And another important thing is all enzymes are proteins, but not all proteins are enzymes. So this is a very important statement you have to remember. All enzymes are proteins, but still there is an exception like ribozyme. It is an enzyme which is made up of RNA, ribozyme. That is the only exception, but other than all the enzymes are proteins. And this ribozyme, for the discovery of this, Venkatraman Ramakrishnan, he got Nobel Prize for this, right? Now, what is the size of enzyme? So usually they will be a globular protein, 3-dimensional shape. So it can have 62 to 2,500 amino acids. Okay. And the molecular mass may range from 12 kilodalton to 1000 kilodalton. We have range of size and shape. So what is the enzyme? What is the mechanism of action of the enzymes? So there are some theories. One is lock and key hypothesis, where you can see here, this violet color one is enzyme and green color one is substrate. So this substrate is exactly complementary to the enzyme shape. And when this binds tightly, there will be reaction which takes place and we'll get the products. So I will just read out. So a substrate is drawn into the active site of an enzyme, and the substrate shape must be compatible with the enzyme's active site in order to fit and get reacted. And the enzyme modifies the substrate, and usually the substrate will broken down and the products will be released. So here, this transition state or intermediate state is called enzyme substrate complex, where the enzyme active site is involved with the substrate. Okay, and this resembles how a lock is fitting into the key. Therefore, it is called as a lock and key hypothesis. And here, enzymes possess specifically shaped active sites for reacting with one specific substrate, thereby generating pure products free from unwanted byproducts. So only it is very specific to only one substrate because of the complementarity. Here, rather than the shape, here you have the amino acids in that site and that will be complementary. Okay. So when it is very specific, there is no chance of getting any byproducts. Only the desired product will be generated. So that is how the mechanism of action of the enzymes. Now, what is the biotechnological process of enzyme production? So how the enzymes are produced? First is screening. First, we have to choose a desired microorganism for the desired enzyme. So not all microorganisms can generate all enzymes. Right? Based on its physiological functions, it will generate. So first, we have to choose the microorganism very carefully. For that, like whatever the enzyme we want to produce. Then modification. So possible application of genetic engineering to improve the microbial strain. So we can do either, so in the first class we have discussed about cloning, all these things. So if we want to produce more amount of microorganisms, we can go for such kind of cloning by specifically isolating a gene and inserting into production of rDNA, recombinant DNA using plasmid or like that, and we can produce more copies or we can modify also. Then laboratory scale, which is called pilot scale. Okay, that means first we have to standardize the things. Okay, once you got microorganisms, now we have to start producing the enzyme. That means the gene has to get expressed. The enzyme has to get expressed. For that, you have to optimize the conditions like pH, temperature, media, everything you have to check. First, you have to provide some temperature, whether the enzyme is producing. Then you have to modify the conditions. And again, you have to check whether the production has increased or decreased or is it same. Like that, we have to check. That is called pilot scale in small scale quantities. Then we should go for the pilot plant. So the previous one is in lab. In the pilot plant, we have to go for the small scale fermenter. Once the standardization has been done, then we should go for the next level, that is pilot scale. If we got succeeded in the pilot level also, then we should go for the industrial fermentation. So there will be very big fermenters where liters of the products will be generated. So why we have to go do all these things before that? Because it actually costs very high. So if we fail in large scale, so it will be loss of money, time and efforts, everything. So before going into that, first we have to ensure that it is successful. All the conditions are perfect. Okay, this is the general process, how the enzyme biotechnology has been done. Now the commercial enzyme production, one example here it is showing. So pectinase is an enzyme. So that has to be, our product of interest is pectinase. So what the scientists will do first, what is the source of the pectinase? So which organisms will generate the pectinase? So they have selected Aspergillus niger, which is a fungus. So from fungus, they have cultured this fungus, Aspergillus niger. So how they will produce this Aspergillus? First, you have to study Aspergillus niger and what is the metabolism that is going on and how it produces this enzyme. So this Aspergillus niger produces pectinase as extracellular. So there are intracellular and extracellular. If it is an extracellular product, so it is more convenient because that will be all released into the culture media. So it is easy to isolate. If it is intracellular, you have to kill that particular organism and then you have to isolate. So like that, the process will change based on the extracellular or intracellular production. Now, this information you know. Now, so first of all, what is a pectin? What is a use? Why we need to do? It's an insoluble substance found in the cell wall of plants. Of course, in the fungi also. So this helps, this is useful in drinks industry where juice, fruit juices, they will become cloudy. So further, because of presence of pectin. Now this pectinase is used to reduce the, suppose you buy a fruit juice from a shop, it is very cloudy. Will you take? No, obviously we think that some contamination is there or some outdated juice. So what the business people they will do, first they have to reduce this cloudiness of the juice. So first, why this cloudiness is because of pectin. So if we use the pectinase, that will degrade all this pectin and we'll get a clear juice. Okay, we think that, okay, it's very fresh and the consumer will buy. So therefore, this pectinase is very much useful in the juice industry, fruit juice industry. So therefore, the effect of pectinase is to clarify the fruit juices and make it flow more freely. So this is one kind of example, commercial production of the enzymes. And what are the other properties of enzymes? Where exactly they can use? Control ripening in the fruits, and then fruits or vegetables. Then cause food spoilage. So these enzymes are also involved in rottening of fruits and vegetables. So for food spoilage, suppose if you want to stop food spoilage, we can stop the enzyme or we can increase the production of the enzyme. So accordingly, we can modify. Then it is responsible for the changes in the flavor, color, texture, and nutritional properties. And also the enzymes can get inactivated when we heat to the extended storage of the foods. So that means enzymes are proteins. So they will get denatured. Not all proteins are heat resistant. So some enzymes will get degraded. And they control oxidation and spoilage. That is in the bioconservation. Then increase nutritive value in the form of phytase, proteases, and in fermentation and purification, extraction. So these are some of the properties of enzymes, and in these industries, the enzymes will be more useful. Okay, so now what are these? This is about the application where we can use. Now, what are the sources of the enzymes? There are three major sources of the enzymes. First, plants like papain from papaya, pineapple, okay, bromeliad, and then animals. From animals, we can get from rennet, from cows, then microorganisms. So more than 80% of the enzymes we will get from the microorganisms like bacteria, yeast, and fungi. So vegetable coagulants. So vegetable coagulants are nothing but the plant extracts which contains proteolytic enzymes. Coagulant means it is nothing but a clumping of proteins. So whenever the protein break down, that will form a clump, and that forms a coagulant. Suppose the best example is milk. Milk contains protein, casein. So whenever in our houses, like if we just add some lemon juice to the milk, what happens? Immediately, all the proteins, which are like casein protein in the milk, that will broken down. And what happens? That will get accumulated at the bottom. So from which we can prepare actually paneer, all these things. So that means the protein will get coagulated because of proteolytic enzymes. So this proteolytic enzymes can actually be used in the cheese industry and even like paneer industry. That is food industry where we can do. The next is, so these vegetable coagulants are used primarily in products where specific flavors and consistency are desired, and or in remote regions where these coagulants are unavailable. So if some regions will be there where these coagulants won't be available. So in such, like not for the household things, mostly for the restaurants or big hotels, okay, like that, like in the large scale where they are not present. So from there, actually we can export. Or if the flavors and consistency is required for any specific thing, that also where we can use this vegetable coagulants. And the extracts of cardo flower used in Spain and Portugal for the production of some DOP cheeses. Okay, so if you suppose like in some areas they'll have some local food products. Okay, they will use some material to produce some kind of dishes. Suppose if here in India, if we have some Spanish hotel, and obviously people expect the dishes from the Spain. So if they require some kind of coagulants, here they are not available. So they have to get that coagulants from the Spain so that to prepare particular dish. So like that, this industry actually helps in like in the preparation of some food dishes, all these things. So for this cheese, which is DOP cheeses, they are very soft or semi-liquid and maybe spoonable. With spoon we can eat. So the flavor is very strong and bitter. Okay. So to get that flavor, we have to use this extracts of card of liver. Okay. And then extracts of Calotropis procera. So this is another plant which is used in Ghana. So this is used in the production of Vara or Vagasi cheese. This is another type of cheese. It will be hard in texture and high melting point. It is suitable for the grilling. So usually we grilled foods. So this is also used as a grilling. So like that, each area we will have a specific local dishes and we require local flavors for that. So for supply of all these things, this is a very good industry, food industry, where we can use these enzymes. So due to high production costs and low yield and development of off flavors, so vegetable coagulants are not used in the industrial cheese production. Okay, so this is one of the disadvantage. Then the enzymes which are, that is the plant sourced enzymes. Now the enzymes which are extracted from the animals. So we will look into some of the examples. Like lipase. So lipase is an lipolytic enzymes. Lipase means it will degrade the fat. Okay. So they are extracted from the throat of cows, sheep or goats, which enhance cheese flavor. But they are very expensive. Okay. So as soon as the source, if the source is rare, then the cost will be more. And then rennet is another enzyme which is produced from the stomach of cows. And the enzyme chymosin, the same thing, renin, and the another name is chymosin, is obtained from calf stomach, or now recently they have identified in a microorganism also. This renin, it is helpful in the cutting of the casein proteins which are present in the milk. So I told you just now. So into smaller pieces. So they will solidify milk. So it is very much useful in the paneer production. Okay. So enzymes produced by microorganisms. So this also just now I have explained about Taq polymerase. So you can see the image of hot spring. I just explained, like, so the water bodies, but it will have very high temperature. Okay. So they are thermostable enzymes which are essential for PCR reactions, polymerase chain reactions. And they are isolated from hot springs, which are Thermus aquaticus, hot spring dwelling species. Okay. Then another enzyme is invertase. So this is derived from Saccharomyces cerevisiae. This is also called as sucrase. Invertase is also called as sucrase. And this sucrase digests sucrose into glucose and fructose. So we all know in the carbohydrates, it's a sucrose is a disaccharide which consists of glucose and fructose. So this sucrase will digest this sucrose to give glucose and fructose. This is mostly useful in the candies preparation. Okay, like chocolates and all. And most commonly used in the chocolate covered cherries. So the cherries which are covered with the chocolates, those are some specific brands, in that they will use this enzyme. So here we have enzyme classification. So here we have 6 classes of enzymes, if we see. Like what are they? One is hydrolase, isomerases, ligases or polymerases, lyases, oxidoreductases, and transferases. So the hydrolases, they are involved in the hydrolysis reaction. Then isomerase. So you have examples also. Lipase is a hydrolase. Then isomerases, they will rearrange the atoms within a molecule. So examples are phosphoglucoisomerase. Then ligase. Ligase means ligation, joining of two or more chemicals together. Examples are acetyl-CoA synthetase, where it combines acetate and coenzyme A to form acetyl-CoA. Then liases, splitting of a chemical into smaller. Liase means to lyse into smaller parts, but here no water is involved. So examples are fructose 1,6-bisphosphate aldolase. Then oxidoreductase. So they are involved in transfer of electrons or hydrogen atoms from one molecule to another. Lactic acid dehydrogenase. Then transferase. So transferase means transferring of functional groups, moving functional group from one molecule to another. So hexokinase, this is one thing. So like this, all enzymes are classified into any one of the 6 classes of the enzymes. So first, enzyme isolation methods. So first we have to go for the animal origin or plant origin, or if you want microorganism, so we should go for the fermentation. If we are taking from animal or plant, first we have to grind. And from that, after grinding, we have to extract. And if it is a microorganism, it can generate either intracellularly or extracellularly. If it is intracellular, again, you have to go for the disruption. You have to kill the cell and you have to isolate. If it is extracellular, no need of disruption because it will release the enzyme into the media, surrounding media. So whatever it is, finally, we have to filter the product, what we have got, and then we should concentrate the enzymes. So along with this enzyme, we will have some debris also, right? If we disrupt, though we filter it, there can be any other products also. So first we have to concentrate them. Then we have to purify. After purification, we have to dry them. And then we will get the final product. So we can either sell them in the form of a powder form, or if you want to sell them in a liquid form, we have to again resuspend them in an appropriate media, and then we have to like marketize. So it's the same thing, like if it is plant cell or animal organism, we have to do it for the cell disintegration, then liquid solid separation. We have to remove the waste. We have to remove nucleic acids. We have to purify. We have to concentrate, and we have to get the final product. So in addition to the previous applications, some other applications are, they are used in the analytical applications of enzymes, and then animal feed industry. So in animals for feeding, then meat and fish processing industry, dairy industry, leather industry to soften the leather, then CIP and cleaning of microfilters like detergents, and then modification of fats and oils, pulp and paper industry, fruit juice processing industry, then bulk and fine chemical production, and enzyme replacement theory. So in all these industries, so the enzymes are used. Now, already some enzymes are used in the industries. So what are the applications? First is pepine. So pepine, they got it from latex of unripe papaya fruit. Okay. And the action in food, what it will do is, it will do protein hydrolysis. It hydrolyzes the proteins. Therefore, this is used in the meat tenderization industry. Okay. Then next is bromelain. So bromelain was isolated from pineapple juice and stem. So it's importance is in muscle and connective tissue protein hydrolysis. So again, this is in meat tenderization. So when like meat processing, so it involves muscles. Okay, so in that, to tenderize the meat, we can use this papain and bromelain. Then ficin, this is from fig fruit latex. And again, this also in muscle and connective tissue protein hydrolysis. But this is not widely used because it is more costlier than the other two. Okay. Then next is chymosin or rennet, which is isolated from calf, abomasum or abdomen, stomach. So this is the action of this enzyme is kappa casein hydrolysis. So kappa casein proteins are hydrolyzed. That in cheese making for the cheese industry. Then pepsin. This is isolated from bovine abomasum. So this is again involved in casein hydrolysis in cheese. So this also can be used along with the rennet in the industry. The next is lactoperoxidase. This is from, upper one, this is lysozyme, which is from hen egg white. So this is used in the hydrolysis of bacterial cell wall polysaccharides. Therefore, this is used in the prevention of late blowing defects in cheese by spore forming bacteria. Okay. So that means to kill the bacteria in the cheese production. Then lactoperoxidase from cheese whey or bovine colostrum. So the use is, the mechanism is oxidation of thiocyanate ion to bactericidal hypothiocyanate. So it is in cold sterilization of milk. So we have pasteurization, which is heat sterilization. Here we are not heating, but we are sterilizing in the cold conditions. Then aminopeptidase. So this is isolated from Listococcus lactis and Aspergillus species and Rhizopax oryzae. So this releases free amino acids from N-terminus of proteins and peptides, and they releases free amino acids from N-terminus of proteins and peptides. Okay, that is during it is like a denaturation process, or we can remove one by one amino acids from the N-terminus. Then lipase or esterase, which was isolated from gullet of goat and lamb or calf, abomasum and pig pancreas. So the hydrolysis of triglycerides or fat is done. So this is for flavor enhancement in cheese products. So that means mostly in the cheese and dairy industry, so the enzymes are used. And in the food industry, so what are the enzymes? So first, enzymes break down specific components like fruit and vegetables, like pectin, starch, proteins, and cellulose. So which will, when we break down all this, this will result in increased yields, shortening of processing time, and improve sensory characteristics. The taste, flavor, everything will be increased. So some examples of these enzymes are pectinases and cellulases. So they are used in the breakdown of cell walls in the fruits and vegetables. Therefore, the extraction of juice will be increased. Therefore, the yield will increase. Okay. And also this will decrease the viscosity of purees and nectars. And also cloudiness will be reduced and the texture will increase. So normally if we see any fruit juices, if you take at home, like Mosambi or something like that, what happens? It is not similar to the juices which we get in packets, right? It will be cloudy and it will be viscous. Even taste will be somewhat sour, not that taste, not that sweet. Right? If you add more sugar only, you will get the sweetness. But at the same time, if you buy something like the packed foods, processed foods, that will be very clear, very liquidy and it will be very good taste, sweet taste. So that means after processing of everything only you can get such kind of clear fruit juices. So in that industry, usually these enzymes are used. The next is brewing and alcohol production. So the enzymes like cellulases, beta glucanases, alpha amylases, proteases, maltogenic amylases, amyloglucosidase. So these are all the enzymes which are involved in brewing and alcohol production. So all the top ones except amyloglucosidase, they are used in the liquefaction and clarification and to supplement malt enzymes. Whereas amyloglucosidase is involved in the conversion of starch to sugar. Then in the baking industries, we have alpha amylases, which will break down the starch or maltose production. Then amyloglycosidases, that is involved in the saccharification. Then maltogen amylase, which delays the process of, like if the bread becomes stale. So to delay that process, maltogen amylase can be used. Then protease, which will break down the proteins. Then pentosanase. This is involved in the breakdown of pentosan, leading to the reduced gluten production. Then next is in the wine and fruit juices, we use pectinase, glucose oxidases, which will yield for the, and also like juice clarification and oxygen removal. And then meat. In the meat industry, proteases can be used for meat tenderization. Then proteins industry, we use proteases, trypsin, aminopeptidases for breakdown of various components. And then starch industry, we use alpha amylase, glucoamylases, hemicellulases, amylases for the modification and conversion, like from dextrose or high fructose syrups. Then some other enzymes are like, what are the functions of the enzymes? So alpha amylases, they convert the starch to dextrins for production of corn syrup. And they solubilizes the carbohydrate in the barley cereals. Then glucoamylase, they convert the dextrins to glucose. Here also in the production of corn syrup. And also conversion of residual dextrins to fermentable sugars. Then beta glucanases, they are involved in breakdown of glucans in malt and other minerals to aid filtration after mashing for brewing process. Then lipase, so this is involved in the enhancement of the flavor, okay, and shortening of the time for cheese ripening. And we can also improve the quality of the cheese and production of enzyme modified cheese or butter. In that industry also we can use this lipase. Then pepine, it is used as a meat tenderizer for brewing to prevent chill haze formation by digesting proteins. Then chymotrypsin, it is for curdling of the milk to form curd. So by breaking kappa caseins and even in the cheese production. Then microbial proteases, processing of raw plant or animal protein and production of fish meals, meat extract, tenderizing proteins and meat extenders. Then pectinase, it is involved in the treatment of fruit pulp, okay, for in the juice industry. Okay, so these are some of the functions of the enzymes. Then lactase, it is involved in the dairy products, okay, where lactase is it is absent, okay, and it is main function is breakdown of lactose in whey products. And then acetolactate decarboxylase, which is involved in the reduction of maturation time in wine by making or by converting acetolactate into acetone. Then glucose oxidase, It is involved in the conversion of glucose to glucanoic acid to prevent Maillard reaction in products which are caused by the high heat. And then cellulase. It is involved in the conversion of cellulose base to fermentable feedstock for ethanol or single cell proteins. So like that, you have many products or many enzymes which we can use in the various industries. So what are the advancements? So these are common things like direct extraction of enzyme, and we can do it. But we have some advancements also by using nanoparticles. So what are now the nanotechnology or nanoparticles has been applied in almost all the industries. So what are the applications? So first of all, what is nanotechnology? It is nothing but the manipulating the matter in an atomic scale. That means nano means 10 to the power of -9 scale. So we have to like reduce the component into atomic scale or very small size, and we can apply them. So nanotechnology refers to the constructing and engineering of the functional systems at a micro level, or we can call it as atomic level. So a nanometer is 1/1000th of a meter. Okay. Like roughly the width of 3 or 4 atoms, or average the human hair has 25,000 nanometers wide. So you can easily imagine what is 1 nanometer looks like. Our hair width is 25,000 nanometers width. So that means it is very, very, very minute. And first ever concept was presented in 1959 by a physicist, Dr. Richard Feynman. So this nanotechnology is a multidisciplinary branch. So it's not about biology or physics or chemistry. It almost involves all these subjects. And invention of the scanning tunneling microscope in 1981 and the discovery of fullerene in 1985 led to the emergence of nanotechnology. So the nanotechnology, the term was coined by Norio Taniguchi in 1974. So this can be used in the enzyme biotechnology. So the application of nanomaterials as novel supporting material for enzyme immobilization has generated incredible interest in the biotechnology. So what it will do, these nanoparticles, we can make some small meshes or pores or nanofibers so that we can immobilize, we can place the enzymes in one place. That is enzyme immobilization, that is trapping the enzyme. So we can form like nanoparticles, nanofibers, nanotubes, nanopores, nanosheets, or nanocomposites, which will have a high surface to volume ratio. And they can hold a high quantity of enzyme and where the reaction can take place, and reaction kinetics can be facilitated in that particular area, which will improve the biocatalytic efficiency of enzymes in the industrial scale. So the current status of versatile nanomaterial support for biofuel production. So by employing cellulases and lipases, now bioethanol or biodiesel. So all these biofuels are generated because all these conventional sources of energies like petrol, all these are getting exhausted. So we have to look for the alternate sources of fuel and energy. So these are good alternatives. And nanomaterials will become an integral part of sustainable bioenergy production. So how this actually they will do is, so in the fermenter or bioreactor, so we have the pre-treated lignocellulosic or triglyceride or non-food biomass sugars will be present. In that, we have a nanomaterial or nanoparticle along with the enzymes which are bound. So they are immobilized. The enzymes are immobilized on the nanoparticles. So we have to add these immobilized nanoparticles. And now this biomass or sugars will act as a substrate on which these enzymes will start reacting and we will get the products. And that products can directly, we can isolate, we can separate, and we can produce biofuel or ethanol or butanol. That means here, rather than adding an enzyme directly into the substrate, if we immobilize them, it will have a large surface to volume ratio. So more number of enzyme molecules can stay together and the reaction will be faster. So single enzyme nanoparticles means the enzyme led short and brutal lives, because if you directly add the enzyme into the medium, so that can be degraded because the enzyme is again a protein, right? So a team of scientists at Department of Energy Specific Northwest National Laboratory in Richard Caged, they have developed a single enzyme to create a new class of catalysts. They are called single enzyme nanoparticles. So here the nanostructure protects the catalyst, allowing it to remain active for several months. And Kim and Great, who are working in W.R. Ville Environmental Molecular Sciences Laboratory, they modified a common protein splitting enzyme called alpha-chymotrypsin. So to increase the enzyme activity and thermostability, so these nanoparticles are helpful. So study on impaired pectate lyase. So they have isolated this pectate lyase from Macrophomina facilina. Okay, from facilina. So from this organism, they have isolated this pectate lyase in the presence of hydroxyapatite nanoparticles. So they can not only act as a chaperone, but also chaperones are nothing but heat shock proteins normally in our body. So they will give the stability in the form of against the temperature, and also can serve as a synthetic enhancer of activity of an isolated extracellular pectate lyase with low native state activity. And the purified enzyme, it will show activity at 50 degrees centigrade and pH of 5.6. Okay, however, by adding these nanoparticles, it can also show its half-life has increased to 90 degrees centigrade. So that means from 50 to 90 degrees centigrade, the efficiency of the action of the enzyme has been increased by the addition of nanoparticles. Then enzyme metallography. So enzyme metallography is a new biological labeling and staining method which is developed at nanoprobes. So here it uses a targeted enzyme probe with a novel metallographic substrate to provide a quantum leap in staining clarity over conventional chromogenic and fluorescent substrates. So instead of, we have some fluorescence techniques like in situ hybridization or immunohistochemistry. So in that, instead of directly using the probes, we can use this nanoparticle labeled enzyme probes. So for better activity of the fluorescent enzymes. That means we have the fluorescent substrate. So these enzymes will convert the substrates into products and we will get the fluorescence and we can measure that, right? So now we will discuss about a case study. So nanotechnology to harness the natural light produced by fireflies. So we all know like fireflies, they produce light. And that is again because of enzymes which are present. So by designing a way to chemically attach, genetically manipulate, any doubt? Please mute yourself. Yeah. So by designing a way to chemically attach genetically manipulated luciferase. So luciferase is the enzyme which helps in the glowing of the firefly. So here we have to manipulate the genetically manipulated luciferase enzyme, and they are directly attached to the nanorods. So this was done by the scientists at Syracuse University, and this is to harness the natural light which is produced by the fireflies. So this is by the reaction of the luciferin, which is a substrate for the luciferase enzyme. And in May's laboratory, the enzyme is attached to the nanorod surface, and this is added to fireflies again, and this can be used as a fuel actually in the large scale if we can do. So this is one of the case studies. And now the energy that is released when the fuel and the enzyme interaction that is transferred to the nanorods. So therefore now the nanorod starts glowing. Instead of, there is no any organism, fruit fly, but directly they have isolated the enzyme and substrate and they have used a nanorod. So now the nanorod starts glowing, and the process is called bioluminescence resonance energy transfer, which is called BRET, B-R-E-T. And the nanorods are composed of an outer shell of cadmium sulfate and an inner core of cadmium selenide. So outer they are cadmium sulfate and inside it has cadmium selenide, and they have prepared a nanorod. And to that they have added luciferin and luciferase enzyme, and the reaction has carried out. So this is a case study. Now there are various techniques of enzyme immobilization. So here the enzymes are, so I already told you they are biological catalysts, all these things. So they have to first immobilized or insolubilized so that they can get retained in the biochemical reactor for the catalysis. Okay, so it is a process of enhancing the activity of the enzyme and enhancing the half-life of the enzyme. Okay. So enzyme immobilization is a process of confining the enzyme molecules to a solid support over which the substrate is passed and converted to product. And the process thereby involves the movement of enzymes, cells, organelles, etc. in space is completely or severely restricted, resulting in water-insoluble form of enzyme. So therefore, now enzyme is not soluble in water, but it is present on a substrate. So what is an immobilized enzyme? It is the restriction of the movement in space is done in immobilized enzyme. Okay. So what is the need for immobilization? It is for protection from degradation and deactivation and retention of enzyme and enzyme-free products, then recycling and repetitive use. Once, if the enzyme is there in a media, you cannot again isolate the enzyme back. But if it is attached to some nanorod or any substrate, you can again isolate that enzyme, immobilized enzyme, and you can use for the next reaction. So for repetitive use. Excuse me. Then cost efficiency, enhanced stability, and to control releasing agents, and the ability to stop the reaction rapidly by removing the enzyme from the reaction solution. Okay, if you want to remove the enzyme, you can easily do. And it allows the development of multi-enzyme reaction system. That means if you are adding all the enzymes to the media, then there can be no chance of multi-enzyme reaction because they can get mixed up. But if you are using some substrate, you can use different substrates having different enzymes, and you can develop a multi-enzyme reaction system. The features of enzyme immobilization are, so the enzyme phase is called as a carrier phase, which is water insoluble, but it is hydrophilic porous matrix like agarose or cellulose. And the enzyme phase may be in the form of fine particulates or membranous or microcapsules. So the enzyme is in turn bound to another enzyme by cross-linking, and a special module is produced by employing immobilization techniques, through which the fluid or substrate can be passed and you will get the product finally, which can be removed by the reactor. And the support or carrier, which is unutilized in the immobilization technique, is not stable at pH, but if you immobilize them, they can actually be more stable comparatively. So the advantages are we can use for multiple times and repetitive use of single batch of enzymes, and they are more stable. We can stop the reaction anytime, and the product is not contaminated with the enzyme. So purification of the product is also very easy, and easy separation of enzymes from the products allows development of multi-enzyme reaction system and reduces effluent disposal problems. But there are some disadvantages of enzyme immobilization, which involves they give rise to additional cost, okay, for using nanorods or any substrate. And the invariably affects the stability and activity of enzymes. Sometimes that can also interfere with the activity. And the technique may not prove any advantage when one of the substrate is found to be insoluble. When already substrate is insoluble, then the efficiency will reduce because enzyme is also insoluble and substrate is also insoluble. The interaction may reduce. And certain immobilization protocols offer serious problems with respect to diffusion of the substrate into the enzyme. Okay, so you have to choose them carefully, like based on the protocol as well as substrate and enzyme. What are the characteristics? Okay. So the methods of enzyme immobilizations are carrier binding, cross-linking, and entrapment. So in the carrier binding, we can use physical adsorption. We can adsorb the enzyme to any physical substance, or ionic binding to ions if there is any charge to the enzyme. Then covalent bonding, and we can just cross-link the enzyme material using like agarose or something. Then entrapment. So we can use some lattice or microcapsules to entrap the enzymes. Like gelatin, all these can be entrapped. So carrier binding, which is physical adsorption. So this method is based on the physical adsorption of enzyme onto the surface of water-soluble carriers, like ion exchange matrix, porous carbon clay, or hydrous metal oxides, glasses, then aromatic resins. So the bonds between the enzyme and the carrier molecule may be ionic, covalent, hydrogen bond, or coordinate covalent, or even sometimes both 2 or 3 bonds may also be combined. And the immobilization can be brought by coupling an enzyme either to external or internal surface of the carrier. So the external surface binding method is advantageous as it does not involve like pore diffusion, all these things. But the disadvantage is to include exposure of enzymes to microbial attack, and physical aberration of enzymes can happen and turbulence can be formed. The major disadvantage of internal immobilization is pore diffusion. Then methods of immobilization by adsorption are first is static process, dynamic process, reactor loading, and electro deposition. So in the static process, where it is most efficient technique, but it requires maximum time. So here enzyme is immobilized by allowing it to come in contact with the carrier without agitation. It is no agitation is involved. In the dynamic process, we have to admix the enzyme with the carrier, and agitation is required in the shaker. And the reactor loading is by it is involved for commercial production of immobilized enzymes. So here, excuse me, the carrier is placed in the reactor and enzyme solution is added and agitation is given. And electrode deposition is the carrier is placed in the vicinity of an electrode in the enzyme bath and the electric current is passed. Okay. This results in the deposition of enzyme onto the surface of the carrier. So this is how the orange color one is solid support, and this E is enzyme, green color. So these enzymes will go and bind to the surface of this solid support. So the advantages of adsorption are little or no change of enzyme. It is simple and cheap. No reagents are required and applicability is wide. Whereas the disadvantages are desorption of enzyme is it can change the temperature, pH and ionic strength if you want to remove the enzyme again. And it is a slow method. Then another one is covalent binding. So it's widely used method. So where the enzyme is covalently bound to the support. Here the functional groups of the enzyme are covalently bound to the functional groups of the, like what are the support or which is present. So the most common technique is cellulose based support with cyanogen bromide, which is mixed with the enzyme. So the functional groups which are present in the protein which are involved in the covalent coupling are amino group, carboxyl group, phenol ring and indole group. So advantages of covalent coupling are the strength of binding is very strong. You all know like covalent bond is very strong. Leakage of enzyme is little or absent. It is simple, mild and successful technique. Whereas the disadvantages are enzymes are chemically modified and sometimes they can be denatured and small amount of enzymes are immobilized. The next ionic bonding where the enzyme is made to bind to some ionic support like DEA is Cifadex, DEA is cellulose, Dolan 50, excuse me, then carboxymethyl cellulose, then amberlite. So they can be used as a support. Advantages are low cost, regeneration is possible, preparation is easy, and overall enzyme activity is high. And the disadvantage is it is not fit for the industrial process as the efficiency will decrease gradually and the ionic interaction between substrate and enzyme can disrupt the enzyme activity. Then cross-linking, this is based on the formation of covalent bonds between the enzyme molecules and reagents. So it's three-dimensional aggregates will be formed. So it is commonly used and to adsorb enzymes and to stabilize. It prevents the leaking from polyacrylamide gels. The most common reagent used for the cross-linking is glutaraldehyde. So here the free enzyme, upon amination, it will form free aminated protein to which polyethylene glycol, precipitate and salt and solvents will be added and we'll get aminated protein aggregates. And this they are added to glutaraldehyde and finally we will get the cross-linked enzymes, okay, as a bunch like this. So the advantages are very little desorption, enzymes strongly bind, and it is best used in conjugation and other methods. The disadvantages are that it will change significantly the active site of the enzyme. So this is the major disadvantage of cross-linking. Then entrapment. Entrapment is nothing but the enzymes or cells are not directly attached to anything solid, but they are entrapped in a matrix. So entrapment is carried out by mixing the biocatalyst into monomer solution. Now it will form a polymer by changing the temperature and or any chemical reaction. So polyacrylamide, collagen, collagenase acetate or calcium alginate or any other matrix can be added for the entrapment. So the entrapment in gel may cause matrix polymerization, precipitation or coagulation. And in calcium alginate, it is mostly widely used for microbial, animal and plant enzymes or cells. Like if you take glucose oxidase plus polyacrylamide, it will form an entrapped gel with the enzyme. Okay. So types of entrapments are occlusion within a cross-linked gel. So in this entrapment method, a highly cross-linked gel is formed as a result of polymerization, which will form a wiry mesh like cages. So 1 gram of enzyme per gram of gel or fiber can be entrapped. So here synthetic polymers like polyacrylamide and polyvinyl alcohol also, or natural polymers like starch can also be used in this immobilization technique. Another is microencapsulation. Here, the entrapment involves the formation of spherical particle called microcapsule, okay, in which liquid or suspension of biocatalyst is enclosed, which is semi-permeable, okay. And the advantages are loss of enzyme activity upon immobilization is minimized. So activity will be retained. The disadvantages are enzymes can leak into the surrounding media, and mass transfer resistance to substrate and products can happen. That means the movement of products and substrate can be restricted, and substrate cannot diffuse deeply into the gel matrix. So this is all about the enzyme biotechnology. So we have discussed about what are the entrapment methods, how the enzymes can be isolated, how they can be processed, and how they can be entrapped, and how they can be produced in a large quantities in industries, and what are the other applications. So if we are producing in the industry, it has to be there should be some application of that enzyme. So what are the enzymes, and so what are the applications and sources of those enzymes in other industries? Okay. So are you having any doubts? So in the next class, we will continue with another topic. So if you have any doubts, you can ask.
Ma'am, next week onwards, we have practical classes.
Yeah, I think they will update the schedule. I don't know exactly. So we will see accordingly. The schedule will be given now about the theory classes. So accordingly, we can do. Okay. I have no idea. Sorry. If you don't have any doubts, I will just stop the class. Okay. Thank you, everyone.
Thank you, ma'am.
Thank you.